Hello students. So today we are going to learn the second part of chapter 6, plant water relation. In the present slide you can see a simple animation. Towards the left, forest region; towards the right, rainfall in the field. Moving further, you can see what we are going to learn in today's part. Content: plasmolysis, path of water across the root, mechanism of absorption of water, in that passive absorption, active absorption, further translocation of water, in which we will be learning root pressure theory, capillarity theory, cohesion tension theory, and lastly transpirational pull. With relation, you can see on towards the right how the plant takes up water right from the root region, from the soil, passes through the stem and reaches the topmost part, that is leaf, and through the stomata the transpiration taking place. Further, again few images of planting and the stages of growth, and towards the right, watering the plant, which is an essential factor for the growth. Now here again you can see two animation. Towards the left you can see again how the from the root the water getting transpired till the top of the plant through the roots, through the stem, that is xylem and phloem. Xylem, which conducts the water, reaches till the top, that is still the leaf part, and through the stomata transpiration taking place. So here lower water potential. So you can see in the leaf part how the water is transpired, and you can see the H2O molecule over here, and through the stomata water being transpired. Towards the right, path of water across the root, that is epiblemma, up to xylem in the stellar region. So again you can see the section of the plant through which the water which is passing. Even you can see the phloem conducting food in the downward direction. So as we know, xylem which usually conducts in the upward direction, whereas phloem carrying the food from top to bottom. Now coming to the first part, plasmolysis. Now here first point, exosmosis in a living cell when placed in hypotonic solution is called plasmolysis, or you can say breakdown. During plasmolysis, protoplast of cell shrinks and recedes from cell wall. Thus, cell walls become flaccid, means flat. In a plasmolyzed cell, a gap is developed between cell wall and the protoplast. This gap is filled up by the outer solution. In a plasmolyzed cell, TP, what is TP? Target pressure is always zero. When such cell is placed in hypotonic solution, endosmosis occur, making cell turgid. This is called deplasmolysis, that is opposite of plasmolysis. That is when you keep for example seed or raisins in water, it takes up the water and swell up, which is opposite to plasmolysis, that is deplasmolysis. So in a fully turgid cell, TP, that is turgor pressure, which will be equal to osmotic pressure, that is OP. Hence DPD is always 0. So you can see a image over here showing hypotonic solution, isotonic solution and hypotonic solution. Where in hypotonic, as it takes the water, it becomes turgid, whereas in case of isotonic solution, here the water is which is neutral inner solution as well as the outer part, and hypertonic solution which gets plasmolized and the cell becomes flaccid. On towards the right side you can see the animation image of the cell which is getting plasmolized. Now moving further, path of water across the root, that is from epiblemma up to xylem in the stellar region. That is what which I showed the animation image in the third slide. So again you can see here from roots hair to xylem. So you can see various component over here. So I'll just read out the component which will help us to understand the concept much more clearer. Apoplastic root symplastic root, which you can see here with the light red or pinkish color, and symplastic root the blue color. So how it passes through the cells, right? Then further, transmembrane root, the endodermic control entry to the vascular cylinder, that is stella. Then here epidermis, endodermis, vascular cylinder, which is the cortex region, the vessels, that is xylem, the Casparian strip, plasma membrane. Here much more higher magnification, we can see plasma desmata, through which the symplastic pathway takes place, whereas the pathway along the apoplast, Casparian strip, endodermal cell, water moves upward in the vascular cylinder. Coming to the points, water is absorbed by root hair cell through imbibition, further diffusion, and then finally osmosis, sequentially, or it is a stepwise process. Consequently, the cell becomes turgid, its turgor pressure increases, but its DPD value decreases. The immediately adjacent cortical cell inner to it has more DPD value because its OP, that is the osmotic pressure, is more. Therefore, cortical cell wall suck water from the turgid root hair cell. It then becomes turgid. The flaccid root cell now absorb water from the soil. So you can see the stepwise process. The root is turgid initially, so the cortical cell absorb water from the root, which then becomes flaccid. Then in turn the root cell absorb water from the soil, and again it becomes turgid. Water from the turgid cortical cell is sucked by inner cortical cell, and the process goes on. Thus a gradient of suction pressure, that is the DPD, is developed from cells of epiblemma to the cortex of the root. Consequently, water moves rapidly across the root through loosely arranged living cells of cortex, followed by passage of endodermis, and finally the cells of pericycle, that is the protoxylem, is in close proximity with the pericycle. So it goes continuously. So it is a continuous process. Now, moving further, absorption of water being a continuous process, a sort of hydrostatic pressure is developed in the root cells or cells of the root. This is called root pressure. As we saw, from the root cell, it further pass to the cortical cell and it flows continually. So the root cell becomes flaccid, the cortical cell becomes turgid. Then again the root cell absorbs water, it becomes turgid as it takes water from the soil, and it goes on continuously, because of which it creates a hydrostatic pressure, which is nothing but the root pressure. It is due to the root pressure, water from pericycle is not only forced into the xylem but also conducted upwards against the gravity. That's how it keeps on continuously. Path of the water across the root essentially occurs in the way, that is apoplastic and symplast, as we just saw in the previous slide. Pathways: apoplast, symplast. Symplast, again it can be vacuolar or transmembrane. When some amount of water passes across the root through the cell wall and intercellular spaces of cortical cells of root, it is then called apoplast pathway. I repeat this statement, students. When some amount of water passes across the root through the cell wall and the intercellular space of the cortical cells of the root, it is then called as apoplastic pathway. This pathway occurs up to endodermis. So that means the pathway is through the cortical cells, through the intercellular spaces. When water passes across from one living cell through plasma desmata, then it is called as symplast pathway. It is also called as transmembrane pathway. Now students, I think now here by seeing the previous slide, the image and the statement here, now it is very clear that what is apoplastic pathway and symplast pathway. Apoplast, which passes through the intercellular spaces, that is between the cells, whereas symplast through the cells through the plasma desmata. Okay. Further, note the endodermis is characterized by Casparian strip, which we have seen in the previous slide in the image, a suberized layer that forces all to move in symplast in order to enter the vascular system. Moving further, mechanism of absorption of water. Now, how the process takes place? What is the mechanism? Again, you can see here the image, passive absorption of water through root hair. Again, you can see xylem. Here it is PXY means protoxylem and metaxylem, that is MXY. Phloem, passage of cell, soil particles, cytoplasm, vacuole, nucleus, root hair, epiblemma, cortex, endodermis and the pericycle. Once you know the terms, the region, it becomes easier to understand the pathway. So mainly there are two ways of absorption, that is passive absorption and active absorption. Passive absorption, it is the main way of absorbing water through the roots and not by the roots from the soil into the plant. The driving force is transpirational pull and it is thus proceeds through DPD gradient. So in passive absorption, which is the main pathway which water passes through the roots, there is no expenditure of energy, there is no ATP is used as water moves in accordance to the concentration gradient. Hence, it is passive absorption. About 98% of total water absorbed in plants occur passively. So most of the pathway is passive absorption. So passive absorption occurs during daytime when transpiration is in progress. It stops at night when the transpiration stops. So as we know, during daytime the transpiration occurs in presence of sun's light and the heat, and at the night time as there is no sunlight or heat, so the transpiration stops, or we can say the passive absorption stops. Rapid transpiration creates a tension in the xylem vessel due to negative water potential. This tension is transmitted to xylem in the root hairs. Consequently, water is pulled upwards passively. So here the transpiration continues, that is from the root to the various parts of the root into the xylem vessel, and the reason negative water potential, and then further it is transmitted. During passive absorption, no ATP is utilized. As we just read the point earlier, rate of respiration is not affected. In plants, water is mainly absorbed passively. So most of the transpiration which is carried out in plants is passive absorption. Moving further, the second type, that is active absorption. Here, water is absorbed due to activity of roots. Root cells play active role in the absorption of water. The driving force is the root pressure developed in the living cells of the root. So again, active absorption is because of the activity of the roots. Active absorption occurs usually at night time when transpiration stops due to the closure of stomata. As absorption is against the DPD gradient, there is no expenditure of ATP, that is energy, generated through the respiratory activity of the cells. Now we can see passive absorption which takes place during the daytime, that is in presence of sunlight and heat by the transpiration, and it stops during the night time. Vice versa, active absorption takes place at night when transpiration stops. Active absorption may be two kinds, that is osmotic and non-osmotic. Active absorption, so two types, that is osmotic, non-osmotic. So here again I added an image showing the symplastic pathway, that is the movement which takes place through the cell through the plasma desmata. So here apoplastic path, cortex Casparian strip, pericycle, the phloem component. So water and ion absorption and movements in the root by symplastic and apoplastic pathways. Now, moving further, the first type, that is osmotic absorption. Now, Atkins and Priestley in the year 1922 proposed that water is absorbed from soil into xylem of the root according to the osmotic gradient, which as we learned in the beginning, from the soil into the root and through the xylem further. To create osmotic condition, there is an expenditure of energy, but such absorption does not directly require an expenditure of energy. Also, we learned that there is no use of ATP, or there is no expenditure of energy as such. A gradient of dPd develops from the cell of epiblemma to pericycle due to activity of living cells of root. As the process is continuous, a hydrostatic pressure called root pressure is developed in the root cells. So a root pressure is created because of the continuous process. As we learned the term earlier, the hydrostatic pressure. The root pressure forces water from pericycle to xylem and then upwards to the stem, which is again the stepwise process. So here, mechanism of water absorption, where Kramer in 1949 proposed that water is absorbed by the mechanism. So you can see how the water mechanism in plants takes place right from the root hair through the vacuole, soil particles, epidermis. So you can see the pathway how it is continuous further, passing through epiblemma, cortex, then passage of the cell, endodermis, phloem and xylem on the nearby region. So xylem content through which the water passes further. Here the pericycle region. Now, second type, non-osmotic absorption. Again, Kramer and Timen in 1959 proposed this theory. Sometimes water is absorbed from the soil against the concentration gradient. So there will be no dPd, we can say, or the concentration gradient is not considered, or against the gradient. Such absorption requires an expenditure of energy released during the respiration. So poor supply of oxygen retards water absorption. Moreover, low temperature retards water uptake because of decrease in rate of respiration. So here there is a requirement of energy, or expenditure of energy is required. Also, low temperature retards or does not allow the water uptake, or decrease in the rate of respiration. Again, if there is no supply of oxygen or in the absence of oxygen. Use of metabolic inhibitor also retards the rate of respiration and the water uptake as well. So here you can see root hair absorb water and the nutrients from the soil. So a simple image showing the image, that is the magnification of the root, where you can see the air pockets, the water, soil particles, the water, root hair. Further, translocation of water. Here you can see a simple animation where water is absorbed from the root, passing through the stem, that is xylem, and reaching till the top, and through the top region, that is mainly the leaves, the transpiration process taking place. Here again you can see water potential gradient from the roots, osmosis into the root hairs, from there into the xylem by cohesion and adhesion, and further through the leaf, the transpiration, evaporation from stomata, right? What we learned earlier. So first point, the transport of water with dissolved minerals from root to the other aerial parts like stem and leaves against the gravity is called translocation or ascent of sap. So this process, students, takes place against the gravity. So that's why the term translocation or ascent of sap. So it takes place from the root to the other aerial parts, the stem and the leaves, right, till the top region. Translocation of water occurs through the lumen of conducting elements of xylem, tracheids and vessels in all vascular plants. So this is a system taking place in all the vascular plants where the xylem and the xylem components, tracheids and vessels are present. Ringing experiment has proved that xylem is a path of ascent of sap. So the experiment which is carried out, or called as ringing experiment, proved that the xylem is conducted by ascent of sap, or it is a pathway. Several theories have been put forth to explain the mechanism of translocation of water. The theories include vital force theory, relay pump theory, physical force theory, root pressure theory, etc. So we shall consider following three theories. So various theories were put forward to explain this mechanism of translocation of water. Now, moving further, we'll take up the first theory, that is root pressure theory or vital theory. So again you can see a simple image to demonstrate the root pressure. Here a manometer is attached to the uh, flower pot, the stem part where this manometer is attached onto it, and then with the help of a clamp stem, that is a manometer to measure pressure of exodus from the cut stems. So here first point, according to this theory, the activity of living cells of root is responsible for translocation of water. That is, the living cells of root are the reason for translocation of water. J. Priestley proposed this theory when a stem of potted plant is cut few inches above the soil by a sharp knife. Xylem sap is seen flowing or oozing out through the cut end. This exudation at the cut end of the stem is a good proof for the existence of root pressure. So a simple process, by giving a small cut with a sharp knife, we can see the xylem sap flowing out or oozing out. As water absorption by roots is constant and continuous process, a hydrostatic pressure is developed in the living cells of cortex of root. This is termed as root pressure by S. Hales, by another scientist. It is due to root pressure, water along with the dissolved minerals is not only forced into the xylem, but it conducts upwards against the gravity. So we can see not only the passage of water from the root to the various parts into the xylem, but from the xylem as well conducting towards the upward region, that is the branches, the leaves, and then further the transpiration process is carried out. So the root pressure seems to be largely on osmotic phenomenon, and its development is an active process. The value of root pressure is +1 to +2 bars, which is enough to pump water to a height of 10 to 20 meters. So with this simple process of root pressure theory, we can easily identify how the phenomenon takes place, or the osmotic phenomenon is taking place by active process. And as mentioned, +1 to +2 bars, the unit which is just required to move the water or pump the water to a height as much as 20 meters. So the factors like oxygen, moisture, temperature of soil, salt content, etc. also influence the root pressure. So they are equally important for root pressure theory. Further, objections or limitations of root pressure theory. Always students, when there is a theory put forward, there is always a pros and cons, the positive and negative thing, right? So what is the objections over here? So you can see the image which I added over here for the root pressure. That is the limitation. Rate of transpiration is low. Root still collecting minerals. Water is forced from higher pressure in roots to a lower pressure in the leaves. Okay, so here the can see the objections. Although ascent of sap takes place due to root pressure, there are certain objections raised, such as first objection: it is not applicable to plants taller than 20 meters. Students, just now we learned that it can go up to 10 to 20 meters. That means there are plants which may be more than 20 meters. So this particular root pressure cannot be applied to that, right? Then second point, ascent of sap can also occur even in the absence of root system. Now, as we learned in the previous slide, the ascent of sap taking place by the root pressure theory, but without root system, even then the ascent of sap continues. Then what, right? Then third point, root pressure value is almost nearly zero in taller gymnosperm trees. Now, as we know students, gymnosperms, all these species are mostly tall trees, right? So in that case, the root pressure is zero. In actively transpiring plants, no root pressure is developed. So in case of transpirational plants, or whereas active transpiration takes place, the root pressure theory or root pressure is not developed at all. Next, xylem sap under normal condition is under tension. That is, it shows negative hydrostatic pressure or high osmotic pressure. That is in case of normal condition under tension. To sum up, root pressure is not the sole mechanism explaining the ascent of sap in all plants of varying height. So that means what it is mentioned is the root pressure cannot be the only reason or only theory. There may be some other process, whereas in different types of plants, different lengths of plants varying in different heights. Now, so we learned that in the second type, that is capillarity theory or physical force theory. Here again, I added a simple image, capillarity. How if you put a straw in water or in a glass of water, you can see the water moving through the capillary tube, right? Which we try out whenever we drink cold drink or so. Or this simple experiment we might have carried out in the lab or even at home, that is using 5 different colored solution in a glass, connecting it with a tissue paper. We can see how the water mixes and we can see the colors, how it gets translocated or changes, right? So first point, according to this theory, physical forces and dead cells are responsible for ascent of sap. So here what it is told, unlike the previous one, it is a living cell. So here it is a physical force and dead cells are responsible for ascent of sap. This theory was put forth by Boehm in 1863. Students also remember the scientist name in each of the theories who had put forth the theory. Wick dipped in an oil lamp shows capillarity due to which oil is raised upward. Now this particular phenomenon we might have observed home, that is wick dipped in oil lamp when we burn the oil lamp during festivals or even in everyday process, right? The conduction of water in a straw dipped in water is raised to a certain height because of capillarity. The height to which water is raised depends on the diameter of the straw. So the inner diameter of the straw also depends, that water is raised to particular level. If the diameter is more, the movement of water will be less, like in a straw or in a capillary tube, which you might have used in chemistry lab, where the water movement will be much more larger since the diameter is comparatively very less. So capillarity is because of surface tension and forces of cohesion, attraction between like molecules, and adhesion, attraction between unlike molecules. So here the capillarity is the because of the surface tension, that is cohesion and adhesion. Xylem vessel, that is truncated vessel which is lumen, is comparable with straw. Water column exists because of combined cohesive and adhesive forces of water and xylem wall due to capillarity. It is because of capillarity water is raised or conducted upwards against the gravity to a few centimeters only. So here again it shows that the water can be raised or move upwards against the gravity by capillarity as shown in the two images over here. Now again, objections and limitations of this capillarity theory. So here you can see again I added an image for your better understanding. In one of the beaker it is water, second water with wetting agent, then third mercury. So you can see the levels or the upper meniscus over here in the second tube and the third, right? So few important objections. First one, capillarity tube, that is xylem, must be continuously and completely hollow from one end to the other end, but truncates in xylem show closed end walls. So here capillarity theory cannot be accepted. One of the main reason, usually capillary tube is hollow from top to bottom or vice versa, but in xylem the truncate there are closed ends, right? Second, the lower end of capillary tube, that is xylem, must be in direct contact with the soil water. However, there exists a barrier of root cortex between xylem and soil water. So according to capillary theory, it should be in direct contact with the water, but this not so in actual. The root cortex comes between the xylem part and the other component. Third, narrower the capillary tube, greater is the height to which water column is raised. Thus, taller trees should show xylem vessel with narrow bore that is the diameter. However, in nature, the tall trees show xylem vessels having wider bore. Just now, as we learned in the capillary theory, the diameter, if it is smaller, the water is raised to much higher level. If the diameter is broad or bigger, then the level will be raised comparatively less. But in tall trees, in a natural phenomenon, the bore or the diameter is much more larger, right? So again, these three are the objections in case of capillarity theory. Hence, to sum up, capillarity cannot be the sole mechanism to explain ascent of sap in all the plants of varying height. So here, similar to what we learned in the previous theory, here also capillary theory cannot be the only reason for ascent of sap with different length of height or height of plants, because every plant the height may differ, right, from 1 meter to 10 meter to 50 meter and so on. So depending on the height, there may be some other theory along with the capillary theory. Now coming to the third type, cohesion tension theory or transpirational pull theory. So again students, first see the image over here in a tree. You can see transpiration starting from the water uptake from the soil. Root hair, soil particle and water, through which water is taken up through the root hair, and further from the root to the upper part, there is xylem. So cohesion and adhesion xylem, which we again learnt in the earlier theory. Xylem cells, cell wall water molecule cohesion by hydrogen bonding. Then transpiration in the plant. Again we saw a small animation part in the earlier slide. Xylem sap, mesophyll cells, air space with the leaf, stoma, outside air which is present in the surrounding region. So this is presently widely accepted theory explaining ascent of sap in plants. It was put forth by Dixon and Joly in the year 1894. This theory is based on two principles. So students, unlike the earliest theory, this was the most widely accepted theory. So cohesion and adhesion and transpiration. So first point, a strong force of attraction between water molecules is called cohesive force, while a strong force of attraction between water molecules and lignified wall of lumen or xylem vessels is called adhesive force. Due to combined cohesive and adhesive forces, a continuous water column is developed in the xylem right from the root up to the tip of topmost leaf in the plant. So this cohesive force and adhesive force, which we again learned in the first theory, which combined together attracts the water molecule through the lignified wall of the lumen of the xylem vessel. And further, this water column which is developed is continuous right from bottom till the top part of the xylem or of the plant. Further, the transpirational pull. The transpirational pull developed in the leaf vessel is transmitted down to root and thus accounts for ascent of sap. So it is a transverse pull which is developed from the leaf, which goes till down till the root region. Excess water is lost in the form of vapor, mainly through the stomata found on the leaf. This water loss increases the DPD of mesophyll cells. These cells withdraw water untimely or ultimately from xylem into the leaf. So right from the bottom, the uh, DPD or the diffusion pressure deficit, which continues right from the bottom region till the continues with the xylem till the leaf region. In other words, due to continuous transpiration, a gradient of suction pressure, that is the DPD, is developed right from the guard cells up to the xylem in the leaf. This will create a tension, that is a negative pull or the transpirational pull in the xylem. Consequently, water column is pulled out of xylem. Thus, water is pulled upwardly passively against the gravity, leading to ascent of sap. So this transpirational pull takes place right from the top of the leaf or right from the leaf till the base, that is from the till the root region. So here you can see two images, one a picture image showing the transpiration, the water outlet through the stomata of the leaf, the water in the root which is absorbed further, water molecules pull each other which forms a continuous column till the top of the leaf. Here students, you can see a simple animation showing water molecule, xylem, the cohesion and adhesion which is taking place. Okay, now objection and limitations of the transpirational theory similar to the earlier one. So the transpirational pull to operate, water column should be unbroken and continuous. However, due to temperature fluctuation during day and night, gas bubbles may enter in the water column, breaking the continuity. So the one of the question, now there may be gas bubbles formed because of fluctuation in the daytime nighttime, so there may be some breakup in between, or there may be discontinuous water column. It will be not continuous as such in case of transpirational pull theory. Second, this mechanism assumes that tracheids are more efficient than vessels as their end walls support water column. However, vessels are more evolved than tracheids and are more efficient. So here another thing is that tracheids are more efficient than vessels, which helps to support the water column, which maintains a continuity. But the vessels are actually much more evolved than the tracheids, which is actually seen and more efficient. Third, if the plant is smeared with Vaseline in order to stop transpiration, even then the ascent of sap continues or occurs. Now if you apply Vaseline onto the leaf, as we carry out experiments in the lab, which shows the transpiration does not take place or stops the transpiration or reduces, right? But this ascent of sap, still it is continuous. Fourth, ascent of sap also occurs in deciduous plants that have shed all their leaves. Now students, you might have seen a certain temperature, certain region or certain time period, the leaves are shed off from the plants. Even then the ascent of sap takes place. Then how then transpiration is carried out? That is again a question, right? So these observations point to the fact that besides physical forces, activity of living cells seem to be necessary for lifting the water column up. That means the living cells even carry out the process of lifting the water through the water column. So here again I added two images to understand the concept, that is absorption over here from the root. Further through the xylem, you can see epidermis, the water molecule passing through the xylem component. You can see the pith in the surrounding region, the trunk of the tree, then the cohesion, further the transpiration which takes place through the leaf. Here again another image showing the transpirational pull, the lower epidermis, the mesophyll cells, the upper epidermis and the cuticle. Okay. So students, this is the assignment questions of 1 mark, 2 mark and 3 or 4 mark question based on what we have learned today. So thank you all of you students.